So all this semiconductor chip for your smartphone computer using progressive photolithography from UV light to deep UV, and then so now those, for example, TSMC in Taiwan, Samsung, Intel, they try to push this kind of thing to what? Extreme UV, so that this is a practical technology has been gradually moving from some micron to nano, right? So that like TSMC already make a 5 nanometer FinFET transistor, right? For Apple, the iPhone, and also for this kind of new MacBook M1 chip, so that of course contact photolithography is a 1-to-1. Why doing that? Contact photolithography is a 1-to-1 thing, so that our wafer will in direct contact with our coated this kind of silicon, and then so that in this case, so the key idea of this part is we try to make sure that we have a unique hybrid fabrication, right? Patterning fabrication, so that we can make so this contact photolithography for what? To making the channel, microfluidic channel, and projection photolithography. This one for what? Make some micron or even can go into nano structure, right? So that all the smartphone, like my iPhone 10 is 7 nanometer. iPhone another new iPhone 12, I think it's about 5 nanometer. So that I think this can be really going to from some micron to nano. Of course, this is almost 15 years ago. I used ASML. I should put down SML is only the #1 company in Netherlands. I use ASML 5000 to in my our UST cleaning facility to the minimum feature size. The minimum feature size is actually 0.5 micron, but we actually use this kind of old projection photolithography system to make this kind of even smaller, this kind of feature. So do you understand? So that even though your machine is old and the minimum feature is 0.5 micron, but still we can make much smaller. So this is something is important for all of you to understand, so that how we can really push technology with some combination of this kind of micro and nano fabrication process. So I don't know if you have idea how can we overcome the barrier. I remember at least in this course, you must learn some of this kind of photolithography and micro nano fabrication process. Do you have idea how can we go this micro to nano fabrication? How can we make this kind of smaller micro and nano structure using this all this kind of machine, all the photolithography system? I don't know anyone I believe that in this particular course, by probably the other lecture, they probably also study this kind of photolithography. So do you any idea how can we do? No one can answer. Again, one important thing is we leverage some of fabrication process. When we do some patterning, right? We use photoresist to patterning, right? And then one important thing is we can use some kind of undercut. Now, kissing here, undercut to once you pattern this kind of structure. Let me just show you one. This is very interesting and important thing you should know, right? But if it is a second wafer, right, and then we can pattern this kind of structure, okay, or this kind of this photoresist, we pattern this, huh? Well, first we do spin coating or on this kind of photoresist on a wafer, right? And then we use optical mask and paste some UV light, deep UV, extreme UV light patterning, right? So that in this case, although you can see, when we use this old machine, 0.5 micron, okay, this is 0.5 micron, right? And then, but that's limitation, right? So this is this is photoresist. If you don't think in more a new idea, then you are stuck about this machine, 0.5 micron. But if you have some deeper understanding about microfabrication process, then you can overcome the thing to make it from 0.5 to even smaller. So the key, what's the key? As I mentioned, one of the key things here is undercut. So we can, in many microfabrication process in semiconductor or micro machine, so that we can use some plasma, especially this kind of O2 plasma, so that we can try to use O2 plasma. Now, let me try to put down again. It's one of the very important. We can use this kind of O2 plasma to after this kind of photolithography patterning to do etch this kind of photo PR, and then doing this kind of dry etching, so that they can do this is dry etching, so that in this case we are able to etch away some of these kind of photos, and at the same time they also etch away some of this part. So eventually, after this kind of additional patterning and dry etching, so that eventually we can actually we can really try to make it much smaller. You see, in this case, we can make it sound even smaller feature here. And this is the one we mentioned. This is undercut, undercut, I mean, you etch using this plasma, auto plasma etch away on the surface of photoresist, but at the same time, they also have this kind of plasma can also attack on the side wall of this one. So that is the thing why now 15 years ago, I work with my PhD student, and we can try to make this kind of some micron, 0.3 micron array. And of course, we can also try to push technology, this one to S ratio. So this kind of diameter to this length, S ratio here can go to 51. You know, at that time, 15 years ago, when we do this one, and then we make a breakthrough, so that the company we bought this deep IE machine from STS in UK actually found this parameter is even better than the one they put in the data sheet of their product. So basically, we really understand. I use STS ICP deep IE machine to demonstrate if a good understanding about this microfabrication, you are able to make even much better this kind of micro and nano structure. But of course, this idea of this kind using this kind of plasma etching to do undercut to make some much smaller this kind of device is not new. Okay, so that for example, like there's one professor in UC Berkeley has used this idea to make a FinFET transistor like in UC Berkeley, right? So that this is not new in semiconductor, but this is new in this kind of MEMS micro machine or this microfluidic device. So that that's why I told you this is something I just let you know that for all this fabrication process, there's always some room for you to come with some new idea to continue to push this technology. Okay, so that again, I don't know. Do you have any question? Do you have any question about this particular thing? So that this is actually about this the idea behind of this device, huh? It's really I was my peer student to come with this kind of research, and then we and eventually the STS company in UK actually request about the SEM picture of this one, because we demonstrate we can make it even better than what they can make this kind of thing in their product. So that's something I just let you know. If you're good understanding, then really important, right? So that understand the fundamental concept of microfabrication process can really help you to make a much better device. That's why you can see we can use contact photolithography mesh aligner to do this kind of micro channel, and then use stepper projection photolithography to do this thing. So that is something important for all of you. Of course, I do think that I don't know if you in the lecture that the other lecture also mentioned about this kind of extreme UV photolithography. So that at least, of course, extreme UV lithography is so expensive. One machine. Do you know how much? I see we use ASM 5000, right? It is a minimum feature 0.5 micron, but for most advanced ASML extreme UV lithography, do you know how much is this one machine? Huh? How much is this? No, advanced. The one we're using, this one is old machine, as I mentioned. So the ASM 5000 is very old machine. 5000 is an old machine. We can 0.5 micron. But if you look at ASML extreme UV, so to make a 7 nanometer or 5 nanometer transistor, FinFET transistor, any idea? How much? How much is this machine? Hello? Can anyone guess the number? How much is one SML UV system? Extreme UV system? Huh? Extreme UV. Any idea? EUV. Hello? I don't know if anyone can guess a number. How much? I hope that you can really learn something, right? So that can you anyone understand this kind of real industry, right? So how much is this kind of one SML the extreme UV system? Anyone can guess a number? Hello? I hope that you can really have some more idea about the thing. Anyone? No? No one want to guess this kind of thing? How much? Huh? How much is this kind of hello? Can anyone guess one extreme UV the price? Hello. I don't know if no one can guess. Come on, anyone can guess this one? How much? One machine. Let me show you. One machine is around 100 million US dollar. Okay, so that it's really super expensive. So that it's at this moment I don't think that means I put down specific from the TSMC, right? So each one it's about $120 million. Oh my God, So one machine, let me put down here, so that this is something that's why it's not too many companies in the world can afford. Why? This each is cost about US dollar. It's US dollar 120 million. Okay, this is a super expensive. So at the moment, like TSMC in Taiwan, bought around 50 to 60 machine. This one to do the mass production of this kind of exchange. This for Apple, iPhone, Mac, for all these things, right? So even Intel probably want to place order in the United States because they cannot going to make the 7 nanometer, 5 nanometer, right? So that's of course the next year TSMC we're going to make a 3 nanometer fab in mass production. So this is a very challenging, but at least some require a lot of hard working to push this kind of micro and nano fabrication process. Okay, I just let you have more idea about the thing, so that of course, as I mentioned, DBI is not only for micro fluidics, and also it's important for semiconductor industry, right? So that all this kind of leading semiconductor company are using DBI for TSV, so 3D IC integration. So this become one of very critical in after the Moore's law has reached the end. So that I don't think in the near future our this kind of leading semiconductor can make 5 to 3 nanometers. So that's here wall. So how to go from three-dimensional fabrication become one important thing. Yeah. So that for actually more than 20 years ago, I have been doing all this kind of TSV for microfluidic device. You see, and then of course, I was mentioned one important thing here. Once you can do this kind of micro and nano fabrication, you can really try to make some interesting this kind of electrophoresis chip, right? So we demonstrate with this kind of some microcomputer array. Again, you can replace the sieving matrix. Traditional capillary electrophoresis, right? We have inject some gel, right, as sieving matrix, so that we can make sure that different kind of small and large DMA have different electrophoretic mobility, right? But to inject the gel, very troublesome, very complicated. So that if you can do this kind of micro nanostructure, then we can make this simple, simple and simplify this kind of experiment. Yeah. And of course, we also mentioned we can really do all this kind of separation is even smaller DNA, right? So this is using enzyme to digest lambda DNA, so that we can also demonstrate not only can separate very large DNA molecule. Lambda DNA is is about 48 kilobase pair. Okay, so that and then T4 DNA. So that's also something if you are in bioengineering, right? So that the lambda DNA is about 48 kilobase pair. Let me put down, right? So that the T4 is about 100 kilobase pair. Okay, so that let me see. So lambda DNA is about 48 kilobase pair. So do you know what's the actual length in this lambda DNA? It's on the order, I would say 20, on the order of 20 micron. So actually you can calculate, right? So that the one base pair, right, is about 3.4 angstrom. So we can easily calculate about what's the typical size of the DNA, right? So that, and then T4 DNA, I think is about 100 kilo. It's a very large biomolecule. Okay, so the T4 you can calculate easily about this thing, right? So that basically, I just let you know that it's important for you to have some basic idea about this basic biology for this microfluidic device, right? So that 48. Yeah. Okay. So anyway, so this give you some basic information, so that of course we can see from very large DNA molecule to smaller, so that that show you this kind of technology has some interesting way, right? Yeah. Because if you use traditional electrophoresis, usually there's certain limitation. Most likely you cannot separate very large DNA molecule for traditional it's kind of a gel electrophoresis. And also last time we also mentioned electrowetting. So I think this part we just show you the one of important thing, right? Again, so we should emphasize that this particular one, I think is important, especially for this kind of COVID-19 pandemic. So that as mentioned that this is potentially really can be used for this kind of COVID-19 pandemics, right? So that if if we are able to try to do this kind of again, the idea of this device is leverage electrowetting device, right? Give you electric field and to manipulate the droplet using this kind of surface tension, and then we try to, of course, use an additional microfilter to filter those kind of microparticle sustained in the air. Microparticle, airborne particle can be COVID-19 virus, SARS virus, or other bacteria. So that is something. And then and use this kind of voltage to collect those kind of particle, airborne maybe virus chip on the filter. So this is something I would say is important, useful, especially in this kind of COVID-19 pandemic. Okay, so that of course in the past 1 year, I do think there's a lot of this kind of company and also the research lab try to come with a series of the microfluidic device, a portable thing for rapid detection of this COVID-19 virus. So that, for example, 1 week ago, I found there's one Mexico group try to make this kind of open source Arduino based this kind of device to it's about $30 can really try to make this kind of rapid detection of COVID-19 in about 3 minutes using isothermal, this kind of amplification of the DNA, right, or RNA. So that's something to understand. So in recent 1 year, I think there's a lot of research lab, also company try to make this kind of different types of portable this device for detection of COVID-19, right? Okay, and again, so also mentioned that this is another important thing I mentioned. Genmark is a company near the San Diego, so that they really make this real product based on this electrowetting device plus electrochemical sensor, right? So that this is something, as I mentioned, so electrowetting, it has been exist for more than 20 or 30 years, and so that they gradually make a real product. Also, we'll mention electrowetting based display and zoom lens, right? And also liquid lens for smartphone. Okay. Anyway, so this is something important for all of you to understand, so that all this microfluidic bio chip become more and more important, and for all kind of biomedical area. And last time we also mentioned, right, so that traditional mass spec is very big, very expensive, almost $1 million. So that how can we make a low cost microfluidic or micro machine, this kind of device, right? Not only for DNA, for cell, right, but also for this kind of protein and things. So that I think mass spec has been widely used in biomedical research and drug research. So that and the key thing here is the traditional mass spec, they need this electron spray ionization. Basically, you have some kind of a glass capillary and then some particle filter, and you let this kind of your sample go through, and then apply very high voltage to do what? Electron spray ionization. So that that is some important. That means you create some of the analyze the kind of your bio sample, right? So this is quite important thing. ESI, basically electro spray ionization. So that this is something that means once you have some bio sample, maybe protein, you try to make some charge, right? Ionization, and then let it go through here, and then in this kind of vacuum condition, and then they use this one to do this kind of analysis, right? Spectroscopy, right? To look at the result. And of course, all this thing, this kind of sprays depends on the flow rate, voltage, and distance. So that so eventually near this kind of tip, they form this kind of interesting that telecoin, right? So this is some kind of quite important, I would say, fluid mechanics come with coupled with this kind of electrical field, so that they can really create some unique this kind of analyze this kind of particle for this kind of mass spec. Of course, if you can make a micro machine, how can we make better? Definitely, we might want to make a smaller of this microfluid, the channel for this kind of ESI, so that potentially I think we may able to make using low voltage, right? This is traditional one, maybe 1,000 volt, but if you make a smaller, and then we may use much smaller voltage, low power, to create this kind of ESI in our microfluid channel with this kind of thing, become low voltage, right? So that is something is quite important. You can also look at some of reference. For example, this is from Professor Y.C. Tai at Caltech in United States, so that they really demonstrate. Of course, this area people have demonstrate, like you can separate. Most likely, one important area is to look for those protein analysis is in some of the jar or molecule, small molecule. Yeah, okay, and so that anyway, so here all this takes time, but at least people can gradually you demonstrate, right, so that you can really to create this kind of micro machine, this kind of mass spec, right? So of course, last time we also mentioned lab on CD. This is another branch, right? So we have mentioned about a series of microfluid device, so that this is another branch of lab on CD. Basically, we have shown you a simple micro channel, and then traditionally you can use the syringe pump, right? And then we also electrokinetic microfluidic, right? I mean, using electro-osmotic flow, electrophoresis tools, and then we also have electrowetting based microfluidics, and then lab on CD use again the driving force is centrifugal force, right? So that this is also quite important. Of course, why this is something useful, because every computer in old day, we all have this kind of CD, DVD, right? So that this is a CD player. CD player is one of a very mature, this kind of a consumer product in your daily life. So that if we can do microfabrication on the CD, right? Compact this CD, then to make a microchannel, and also maybe this kind of separation for mixing, and even you see for last thing, multi is another quite important way for this kind of analysis about small molecules. So that I thought this is something quite important. So there's another branch, especially I think Professor Mark Madu in UC Irvine has done a excellent job in this kind of research. And I remember 7 years ago, I visited Professor Mark Madu, and then I remember he actually licensed his this kind of CD to Samsung in Korea, so that of course Samsung do make this kind of lab on CD the product. So this is something quite interesting and important. So how we can gradually, of course, there's a similar company, Gyro in Europe, and also one more in Germany. They also make this kind of a similar or a little bit different variation of lab on CD. Okay, so that at least for this kind of thing, lab on CD have been useful for a series of this application in microfluidics for biomedical application. So this particular show you they are able to do this kind of protein analysis. Okay. Now, of course, we should try to show another important area for this kind of DNA sequencing, so that I don't know if you have some idea about this area. Definitely sequencing is one of a very important technology, and so that more than 20 years ago, there's a quite expensive to do this kind of DNA sequencing. I still remember it's in quite a multinational project, right? Like US have this kind of national initiative for their DNA sequencing, right? It's a span almost more than I would say over $1 billion to do this kind of human genome project. So that but in the past more than 20 years, so that we tried we try to do how can we decode this the double helix DNA with all this kind of ATCG, so that from our engineering concept, basically DNA is like a program, right, in all the biology or biospecies, so that how can we decode this kind of a program, biological program, right, DNA sequencing, so that in all ways, so that the goal is very simple. How can we come with this kind of complete sequencing of ATCG? And then the key thing here is in old day, there's one of the challenging thing is DNA in like in a cell in humans, so that how big is DNA? In human DNA, typically in, of course, you learn the basic biology. Every cell we have some DNA, right? So typically DNA in the cell, like our mammalian cell, is huge, so that it's difficult to do this kind of DNA from this kind of raw DNA from sample, from cell, from human, from all the biospecies, right? So that in this case, important thing here, in order to do this kind of DNA sequencing, most likely, once you get those DNA from the sample, then we need to do what? Cut long DNA into small segments, and then so that we're able to do this kind of DNA sequence. That is one important thing you need to understand. So in general, our conventional way to do a DNA sequencing to have limitation. We cannot process very long chain DNA molecule, so that that's why we have some sample preparation. And then, of course, once you do this thing, and then you can do traditional way is still using this kind of capillary electrophoresis. Okay, so that that thing is also we mentioned in previous slide. Capillary electrophoresis is traditional way for this kind of DNA sequencing. So that of course detect some ATCG, some special molecule, right? So that with different color, full fluorescent color, so that in this case we're able to do this kind of DNA sequencing, right? Using the optically multi-color fluorescent labeling, so we can try to do this kind of thing, right? And then of course in the past many years, so that there's a different technology people could propose. For example, sensors, this kind of approach, the classic one and 454 by this kind of Dr. Jonathan Rothbard. Okay, it's one. I think this is, I will mention it. This is quite important. It's founded by this company by Dr. Jonathan Rothbard. So that he is the one who create a lot of new technology in DNA sequencing. And then Illumina is another big company. They don't call the kind, actually they call the flow cell. Actually, it's one microfluidic device, and then coded with some DNA probe or something, and then optically they do a DNA sequencing. And helical is another biotech, is another way to do this kind of a single DNA thing. Helical and bio is these are the third generation of DNA sequencing technology using like a nano, okay, zero. I think there's special kind of waveguide so that they can do the single DNA sequencing. So again, so there's a lot of this kind of technology thing. Of course, they also have, I forgot, I should also put down there's additional like ion tolerant. Ion tolerant, and also found from this Dr. Jonathan Roseberg from the same guy now who very innovative to come with all this interesting technology for this sequencing. So that and then of course there's another Oxford nanopore. Oxford nanopore is also, I should also put down one more, and basically this is using the semiconductor sequencing way to do DNA sequencing. Basically, it's using this kind of ISP FET transistor PS sensor, and then to do this DNA sequencing, electrically do the sequencing, right? Illumina previously they only use optical to do these sequencings, right? And then of course one more technology is Oxford nanopore. Basically, this is make some interesting nanopore, and they let the DNA to go through, and then they can try to do also single DNA sequencing with look at this electrochemical signal when DNA go to the pore, and then they can do this kind of DNA sequencing. So this is another quite interesting thing. And of course, Oxford nanopore now actually become more portable, this kind of DNA sequencing machine. So that this is something quite important. So of course, in the past many years, there's a long list of different technology, right? So that of course, one important milestone I would say is so that this kind of DNA sequencing efficiency gradually increase, and so that that's why people try in engineering field. People say, you see, because we have new technology invented, so that we're able to do much high throughput sequencing. So that is something quite important, a milestone. Okay, so that of course, let me just show you engineer approach. So of course, in traditional way, called the Sanger sequencing, so that these people have used for the whole genome sequencing, but the traditional way is too expensive. As I told you, it's spend the National Genome Project. They do the they spend almost 10 to power 9, $1 billion to do the sequencing, expensive, and then so that the how do this kind of next sequencing, so that we can really make low cost sequencing, so that and then of course this gradually, right, people have not only look at the whole genome, but also on RNA sequencing, so that look at something not a DNA, but even look at the RNA. Because why we do RNA sequencing? Because DNA something will based on the basic biology concepts from DNA, RNA, protein, right? RNA is quite close to the real look at the phenotype. If you look at DNA, it's preliminary, right? It's a genotype, so that that has something make close to real. No, this kind of this kind of I would say phenotype, so that is something quite important for biomedical research, right? So of course, because people gradually try to do this DNA sequencing for a lot more application, right? Human microbiome. That means people find with this kind of large scale, high throughput DNA sequencing, we want to understand about biology, right? In a human, we have a lot of bacteria inside your body. So how we can understand this kind of human work with a lot of this kind of bacteria in your body, right? So that there's a lot more this kind of thing. Of course, this DNA sequencing gradually also go another level, epigenomics, epigenome, right? So that we understand, right? So that in old day we think the DNA is like a program, control everything about human behavior, but it turned out it's an environmental impact. on this kind of genome and DNA, so that epigenome, that means it's possible this kind of environment impact or genetic can have impact on the biology of this kind of human health. So that anyway, so in the past, I show you that all this thing in the past 20 years, actually, there's a lot this kind of research R&D work on these different kind of DNA sequencing technology, and then so that of course all the technology gradually going to using micro and nano application process, so that as I mentioned, right, so that we have a micro fluid device, flow cell from Illumina, we have nano technology, nanopore, right, so that thing, and then gradually to make a better low cost, high throughput DNA sequencing and low cost, right? So that the traditional way, as I mentioned, so that there's another thing, right? You see, traditionally you have international this human genome project. So you have spent a huge amount of money, about $1 billion, and then try to create some huge machine to do this kind of DNA sequencing for human genome project. And then so that of course there's another quite famous this kind of biotech researcher, Craig Venter, they have created some Celera, and then they really can speed up this kind of DNA sequencing, right? 1 month can do 1 billion of base pair sequencing, right? So that and then of course in the past, you can see gradually there's a lot more people try to study this kind of DNA sequencing, and then also once we finish the kind of human genome, and then it turns out so that there's large number of DNA in the human genome. It seems like not useful, not junk DNA, junk. So the very small percentage genome, they are quite important for this kind of encode this protein, right? So that of course by doing this kind of human genome big project, internet project, then people find so that our humans genome, and maybe it's large percentage is quite similar to the other species, really like a mouse, right? So of course, once we understand this thing, then we can use mouse as animal model for biomedical research, right? Drug research, and then of course, as mentioned, that this kind of in the past more than 20 years, the cost of DNA sequencing actually decreased exponentially, so that the people try to make this kind of interesting comparison with Moore's law in semiconductor industry, so that so it turns out it actually is really maybe even the cost is exponentially decay a lot. So that's something show its importance. Once you have a huge this kind of cost reduction of this kind of technology, and then what happens? You may change the industry, change the way people study biology or this kind of biomedical research and even medical field, right? So that that's something once reach this kind of thing, right? So that local DNA sequencing really has big impact on medical field, right? I still remember last year, US FDA approved one drug required to do the DNA sequencing for personalized medicine. One of cancer drug approved by US FDA last year. Officially, they demonstrate why one of you need to do this kind of DNA sequencing, and then you to prescribe this kind of cancer drug. So this become one of the things. But in the old days, it's impossible. But once become cheaper, then so that imply you still have something, right? Of course, there's another chart to show you DNA sequencing. Now, if you use traditional way, of course, it's cost reduction here. But another, if you use new technology, so that this kind of thing, actually, the decrease of this price, right? The cost, actually, if it even faster than Moore's law, right? Do you understand? So that's something give you some idea. Moore's law is like every like 20 months, you have this cost down here. But if you have new technology, so that you can see they can continue to reduce the cost for DNA sequencing now per base pair to this thing 0.1. This can use dollar per base pairs, right? So that at this moment I can see it's really critical moment, right? In this year 2020, so that as I mentioned, so that finally, so that the US FDA approve this kind of sequencing can be used for personalized medicine, especially for cancer therapy. So that as I mentioned here, and then of course one additional chart I show you here. You can see, in the old day, more than 20 years ago, it's almost 20 years ago, right? So spend the Human Genome Project spent 11 years, spent 2.7, right? G, right? 10 to the power $9, and then you have new way. This kind of Craig Venter spent 3 years, $100 million, and then 454, 3 months, $1 million, and then ABI, Applied Biosystem, 2 weeks, $60,000, and the Illumina, they use 2,000, much smaller, 40,000, right? And then 2010, of course, it'll be much faster. And then, of course, now as I told you, we have a semiconductor sequenced by ion torrent. We have Oxford nanopore, so that become portable and much low cost. So that at this moment, I can see it can really go to, I would say, $1,000, only $1,000, less than $1,000 range for the DNA sequencing, right? And then from $1,000 to $100, I think it's become almost become affordable for medical application now. Yeah. Again, so now at this moment, I would say two important things is we can see that the Oxford nanopore become quite important, and I think I should also show you some of the video about this kind of DNA sequencing thing wise, especially I would say this is quite important. I don't know, do you have any thing? Let me show you some of this Oxford nanopore become really portable DNA sequencing, right? Just like smartphone in old day, now our mobile phones are huge, but now this kind of thing, right? So that become really portable. I think this can give you quite important thing, right, for real application. Let me show you this kind of thing. Mini ion is a portable. You can plug in to this your laptop computer, do the sequencing. It's not something like what I showed you in previous slide. It's a huge lab and then to do sequencing. So this is something completely different now, right? And compared with what now you see, so that I think this is quite interesting for you to look at the one of nanopore.
Nanopore sequencing enables users to produce ultra-long DNA or RNA reads in real time. The technology works by passing a current across electrically resistant membranes into which protein nanopores are embedded. As DNA or RNA move through the nanopores, they cause disruption in the current, which can be analyzed to determine the sequence of bases. The MinION is a palm-sized sequencing device containing a consumable flow cell to which the sample is added. The flow cell contains a sensor that detects the characteristic nanopore signal as the molecule is analyzed. Each MinION flow cell has 512 nanopore channels available to be sequencing simultaneously. Users have achieved as much as 30 gigabases of sequence data per flow cell, making it ideal for any application, from microbiology, metagenomics, RNA and cDNA analysis, up to whole genome sequencing. The MinION is easy to use and can be connected to a laptop, making it suitable for use in the lab or out in the field. Library preparation is a straightforward process which applies across all devices and can be performed manually with Oxford nanopore kits or with an automated library preparation device. Minnow, the MinION control software, can also be run off a laptop, allowing you to gain real-time insight into the progress of your samples and your sequencing run. Minnow performs local base calling to produce FastQ files that can then be taken on for analysis either through epitome workflows provided by Oxford Nanopore Technologies or individuals' analytical pipelines. The long reads produced using a MinION provide a more unambiguous approach to mapping a DNA or RNA sequence, enabling much simpler assembly. With long reads, you can build a clearer picture when it comes to more complex regions, such as repeats and structural variation, while sequencing directly enables the detection of base modifications and methylation without additional preparation steps. Visit the store to buy today.
Okay, so this is, I think from my point of view, it's quite important to let you understand so that compare with the one we show you about this kind of machine in this big lab, right? So you can see now at this moment, we see this kind of how we can use micro and nano technology to really shrink to the portable DNA sequencing, right? Do you understand? So that something show you once a new technology so that we can really do significant low cost DNA sequencing. So that is something quite important for you to understand. Of course, Illumina as another, I think at this moment, Illumina is another quite important thing I may want to show this kind of introductions slide from the company so that at least you understand the basic thing right from this concept.
Hello and welcome to today's talk about next generation sequencing, which is also known as massively parallel or just deep sequencing. Next generation sequencing has completely revolutionized genome analysis. With next gen sequencing, it is possible to sequence an entire human genome in just one day. We start with the sample preparation. Once we have isolated our DNA, we need to generate smaller fragments. Now all of these DNA fragments will get ligated to certain oligonucleotides. There are two types here. These include sequencing binding sites and also an additional complementary sequence which can then hybridize to the flow cell. First, our DNA double-stranded fragment is denaturated. So in theory, the whole genome should be in fragments and all of the fragments have attached two different types of binding sites on each end. All the DNA fragments are now applied on a chip, the so-called flow cell. And as you can see here, there are two types of complementary sequences, the blue ones and the red ones in our example. The DNA fragments will at this ends hybridize with the flow cell as applied to the plate. To make this easier, we just look at one fragment. The first step here on the flow cell is the DNA amplification. By polymerase chain reaction, the complementary sequence to our single-stranded DNA fragment is synthesized. This process, which we have seen here, is done simultaneously for all DNA fragments on the whole flow cell. Now the DNA is denatured and the single strand, which is not attached to the flow cell's oligonucleotides, gets washed away. The next step is called bridge building. You may have noticed that we have two different types of oligonucleotides here, and the second type of oligo will now also hybridize to one of the sticky oligonucleotides on the plate. This process is called bridge building. The DNA is now amplified again, so a polymerase will synthesize the complementary sequence again. This whole process is called bridge amplification. The DNA is denatured, but now you can see that both fragments stick to the plate. The whole procedure will just repeat now. The DNA fragment will build a bridge again, and again we also have the bridge amplification. That whole process will be repeated for several rounds. And this here is the result. So out of one DNA fragment, we generated multiple copies by PCR. We have reverse and we have forward strands from our copy here. One of these strands here is cleaved now, and sequencing can begin. For sequencing, we just look at one copy now. As mentioned in the beginning, a primer can bind to the oligonucleotides. So this is a sequencing binding site here. Sequencing is now done with specific nucleotides. These nucleotides in this case are fluorescently labeled. Only one of them will fit. In this case, the T. The fluorescently labeled T will then be excited by a laser and the fluorescent signal is obtained. This is done with a color code, so we know that in this case blue is T. In the next case, the A will join. Again, the fluorescently labeled A is excited and we will receive a signal again, this time an A. This whole procedure is done for all of the nucleotides, one by one, and this is how we get the sequence. For simplification purposes, we just looked at one of these copies. Now notice that due to PCR we have several copies, so many light signals are obtained simultaneously. The last step now is the data analysis. So here informatic tools are required. The whole process of next-gen sequencing generated millions and billions of reads. These reads are now overlaid and compared to a reference genome. And by this overlay we can now generate the whole genome sequence. This was it. I thank you very much for watching. I put 2 interesting videos in the video description if you want to have further details about this topic. Otherwise, thanks and see you soon.
Okay, so this gives you, I think, base idea about how this Illumina do their sequencing. Again, the key thing is they use this kind of they call the flow cell, right? And then a flow cell from my point of view is one kind of microfluidic device make on this kind of glass wafer, right? And then of course on the surface there's some special type of DNA primer, and then they call the bridge PCR, right? And do this kind of thing. By do the bridge PCR, then they try to do this kind of sequencing, right? That kind they call SBS, right? I think this is all the common thing for all the DNA sequencing, right? SBS, sequence by synthesis, right? So this is one of the thing, and then they use it kind of optically. They can try to, because they have I think 4 fluorescent labeling for ATCG, so that they can use this kind of thing for DNA sequencing. So that is something I just let you know. This is a quite unique, and then at this moment, Illumina is one of as #1 company now to make a real this DNA sequencing thing. Okay, and of course. There's a lot of companies try to make different kind of technology, but Illumina actually at this moment is #1 in the world. Okay, and of course, as I told you, traditionally they have used glass capillary to the DNA sequencing, and then gradually there's people using some glass wafer to make this kind of a microfluidic, right? Large number of microfabricated this kind of microfluidic device for DNA sequencing. For example, this by this MIT group almost 15 years ago, and then they really try to do this kind of DNA sequence. But gradually, I think this kind of technology become too old and then phase out, right? So that, of course, as mentioned, there's another technology using semiconductor, right? Thing, right? So Illumina is using this kind of optically do this detection for this microfluidic device, and so that ion tolerant is another technology actually from the Jonathan Rosbach here. He's one always come with a new idea, right? So that basically is they try to do this kind of DNA sequencing. This is one of I think one important technology they mentioned. They can really do this kind of using leverage in the past 60 years semiconductor technology do the DNA sequencing, and then so that again the key thing here is they are able to use FET transistor. Basically this is a FET transistor, so that I don't know if you have basic idea about transistor. So that basically transistor is they have source, drain, and gate, right? Base transistor basically is 3-terminal device, right? And then so that the 3-terminal device, and then so that then let me show here, so that they have some channel, right? So that you have a this is a drain and source and gate, so that this is a very basic thing, right? You have a drain terminal, and then you have also have another here source, right? Source terminal, and then you have a gate. Okay, there's another gate, 3 terminal. So so you apply the voltage, right? And then you can create some special channel to let the depletion thing, right? So that you can basically you apply this kind of VGS. Now apply voltage between the gate and source here, and then so create the kind of special depletion now here, so it can create some electrical, and then there's some current ID. That means VGS apply thing, and then and in case you can create some current to flow from drain terminal to source terminal. Right? So this is one of the basic way, right? So that how this kind of transistor work, right? So that it's a basic like semiconductor, and then this kind of FET transistor has been widely used. FET transistor, was it in your computer, CPU, and also the smartphone processor, right? So that was a billion of transistor used in this kind of your eyes, all the this kind of electronics. So that of course the idea of this is so this thing basically they replace the gate by additional this kind of special oxide, so that the typical gate in transistor maybe use silicon dioxide. Typically they use some maybe silicon SiO2, while silicon dioxide for as the gate material, so that if we try to make some new type of oxide which can interact with some of electrochemical, some electrolyte, some ion, and then so that in this case we may use this one the gate, right, as some sensing layer, and then basically idea thing is they use FET as electrochemical sensor to send this kind of pH change during this kind of DNA sequencing process, so that they try to provide this kind of DNTP, right? DNTP, there's 4 kind of DNTP, right? And then with this kind of additional micro B, on the surface of micro B, there's some special kind of the DNA primer, and then do this kind of emergent PCR, so that again, this is a very basic way to explain, right? So basically, it's also micro particle based emergent PCR, and then in this case, during this kind of process, they do this kind of PCR, and during the PCR, they can change this kind of pH, so that the FET sensor ISFET, right? So that basically is from FET, ISFET. So it's ion sensitive FET. Now, way to do this kind of sensor, right? And then we can make a pH sensing. So in case, that means during this kind of basically we have some flow control of DNTP, right? And then so that during this kind of PCR, microparticle emergent PCR, and there's change, pH change. So I can try to pH change turn into the what? Electrical signal through this kind of FE transistor, so that I can use this kind of electrochemical sensor to do this kind of change pH, and pH can relate to ATCG. So This is a very basic way, right? They can do this DNA sequencing. But of course, the good thing here is leverage semiconductor industry. Sorry, your iPhone 10 has a 7 billion transistor. Your iPhone 12 has 11 billion transistors, so that the semiconductor technology can really make a large number of this FE transistor. Now, so if you leverage this kind of semiconductor, in the past 60 years, we can reach high throughput, right, DNA sequencing. So that is one of a unique idea of this kind of technology. So that, of course, I should also give you some, maybe one simple this video about how this kind of ion torrent, right, to do this kind of DNA sequencing. So again, so this is something is quite unique, and I would say very special, no, to do this kind of sequencing compared with
sequencing is a powerful technology which can interrogate many targets at the same time, from just a few genes to all of the individual nucleotides in a whole genome. Ion torrent technology from Thermo Fisher Scientific takes an entirely new approach to next-generation sequencing, making it massively scalable, faster, simpler, and more affordable than ever before. The sequencing of DNA is done using a semiconductor chip, similar to the chip found in your digital camera. While the chip in your digital camera has a sensing layer covered with millions of pixels that translate light into digital information, an ion chip has millions of wells covering those pixels. These wells capture chemical information from DNA sequencing and translate it into digital information, or base calls. The sequencing process starts when a sample of DNA is cut up into millions of fragments. Each fragment then attaches to its own bead and is copied until it covers the bead. This automated process covers millions of beads with millions of different fragments. These beads then flow across the chip, each depositing into a well. Then the chip is flooded with one of the 4 DNA nucleotides. Whenever a nucleotide is incorporated into a single strand of DNA, a hydrogen ion is released. This is how the ion torrent system sequences DNA. The release of the hydrogen ion changes the pH of the solution in the well. An ion-sensitive layer beneath the well measures that change in pH and converts it to voltage. This voltage change is recorded, indicating that the nucleotide was incorporated and a base was called. In essence, each well works as the world's smallest pH meter. The process is repeated every 15 seconds with a different nucleotide washing over the chip. For example, cytosine. A polymerase incorporates the C nucleotide in the DNA strand if a complementary G molecule is present. If the nucleotide is not complementary to the next base, no ion is released, no voltage change is recorded, and no base is called. If there are 2 identical bases next to each other, 2 nucleotides are incorporated, the voltage doubles, and the chip will record 2 identical bases called. This process happens simultaneously in millions of wells. That's why it's often described as massive parallel sequencing. The ion chips help you scale the workflow to your research needs so that you can run both small and large-scale projects without the need to change platforms. And the semiconductor approach helps you implement a significantly faster NGS workflow. There are many possible research applications with NGS, from whole genome sequencing to identify association of genomic variations of different diseases, to targeted NGS, where the whole power of NGS is used to interrogate a defined number of targets with possible significance in the pathogenesis. Ion torrent technology from Thermo Fisher Scientific harnesses the power of targeted NGS to make sequencing faster, more scalable, and more accessible than ever before.
Okay, so this just give you, I think with video, it's much easier to show you this idea of this technology. So that again, the key thing here is this is really integration of this semiconductor electrochemical sensor, right, as fact to measure pH, right, during this kind of PCR, imagine PCR using microB. So there's a proton generate, right? And then you can change pH. So that thing, of course, in this case, you see, you're not only use traditional semiconductor process, make FE transistor, but you need a special kind of sensing layer, oxide. Most likely, I think they use some special tantalum pentoxide, so that not the silicon dioxide, so to increase the performance of their device. So that is something. Let me show you. So it's one important thing for you to think about is they really try to show some important thing, right? So you need to have good understanding about the integration about semiconductor FET transistor fabrication with micro machine, this kind of micro chamber, and also you have this kind of a special kind of emergent PCR. Emergent PCR is you have a microparticle with this kind of DNA primers that can do this kind of PCR, right? So that in this case, a good thing here is semiconductor are able to do the high throughput, right? I mean, you have a million or billion of transistors, so that is much easier to do this kind of thing, right? They also demonstrate, right, in this Nature paper, they can really use electrically detect this signal and then try to do a DNA sequencing. Okay, so that is something I show you. This quite amazing technology, and then you can see because semiconductor is technology is easily scaling to from, you see, 1.2 million of this FET sensor, peer sensor, to 150 million of this kind of sensor using semiconductor process. So that is something you can see it can really easily do the mass production, right? So that of course this kind of technology has been used not only for DNA sequencing, for electrophoresis, right? Also, people also try to make this kind of cell chip, right? So you can use, people continue to make interesting micro biomed device to do not only DNA cell, right? So you can do a lot this kind of integration, right? I think so that the micro petri dish you can really manipulate or do 1 million cell on the chip, so that something again. So this kind of biomens microfluidics gradually, I think, become more and more mature, right? You can see in the past 100, 200 years, so that there's a series of technology try to use, right? In the old day, right? Gradually we have industrial revolution, right? So that then railroad steam engine, and then automobile internal combustion engine, automobile industry, computer, semiconductor, and all the semiconductor with MEMS, micro machine, or even nano fabrication, nanopore, so that you can see we are in this transition. So gradually we enter this kind of new technology, not only semiconductor combination, semiconductor biotech, so that you can see I show you a series microfluidic device can use for this kind of or nano application about technology nanopore, right? Can really do this kind of interesting way on this biomens, even nano, this kind of nanopore technology. Okay, anyway, this video I continue to grow, and I show you again, we have brief introduction of micro machine and microfluidics, and show you a series of this kind of basic history, and a series of branch of microfluidic device from electrophoresis and electrokinetics, electrowetting base, and then also show you additional this kind of a branch in microfluidic and nano for DNA sequencing. And then of course microfluidic has become the real product, right? So that's something I have to tell you. This area continue to grow, right? I don't do any question for this thing. I'm sorry, I should go to another important lectures. So that do you have any question for this lecture? No. I think we have to quickly go through another lecture. No, it relate to this kind of one branch, right? For this kind of cell devices, something. Let me quickly go through that introduction slide, so that do you have any question? Any question for this particular lecture? No. I need to quickly show another important lecture about this kind of introduction to this kind of microfluidic device for cell. Any question? So this is another research topic I think I have done. And then again, the important thing is I have some new technology which can really become one of I think quite unique as a way. In my lab at Hong Kong UST, we make a new type of a personalized microfluidic device I call PMEF, microfluidic electrofiltration, for this kind of capture ultra low concentration of cancer cell and CTC for application. Again, this is another big area. So that in the past 10, 20 years, there's a series of microfluidic devices have been developed for this kind of isolation detection of the CTC, circulation tumor cell or cancer cell. Again, the problem is typical. Once patient has cancer, and then normally some of tumor, right, will have leak into the blood vessel, and then CTC, and try to this kind of attack different kind of organ, right? So that this thing, and then again, this is something, the concentration of CTC in human blood is very small, right? So it's about 1 in 1 billion of the cell in human blood. So technology-wise, very difficult. So at least traditionally, there's a different way. For example, biochemically, we have, if there's some spatial biomarker to capture those kind of spatial cancer cell CTC, then biochemically, of course, there's a way people try to do this thing to capture those cancer cell and CTC, circulation tumor cell. And of course, another branch by physical way, so that, for example, people know that some of cancer cell on CTC, they are much bigger than the non-cancer cell. How can we do microfiltration? And for my lab, we try to do a large number of research or different kind of cancer cell, and then by doing a preliminary study and introduce this kind of mechanics, so that we do large number of experiment with different kind of cancer cell, and then using dimensional analysis in fluid mechanics, so that we confirm with interesting new parameter, right? We call the CA, electrochemical number. Basically, is a ratio of two forces, viscous force divided by cell electric force, spring force, so that we demonstrate that CA is quite critical, right? If a low CA, so most cell will stay on our microfiltration device. If a large CA, most cell will have a large deformation, and then most cell will go through. And we discover some interesting CA, right? If find the critical CA, optimized CA number, so we can let the cancer cell to stay and let the non-cancer go through. And then, of course, we also do dimensional analysis to identify this kind of important thing for this kind of thing, right? So that again, the key thing here is we found some interesting way to do things, and also create some model, nonlinear MCK, mass spring damper model, to describe this kind of a nonlinear behavior about how different cell, we are able to show this kind of CA is nonlinear for different thing transition, right? If a small CA, large CA, and then there's very different kind of behavior. It's nonlinear. So of course, that's why we need to use additional nonlinear mechanics to describe large deformation of cell based on this thing, right? So that of course we demonstrate with different kind of cancer cell is behavior. We always identify some critical CA. CA can really optimize this kind of capture efficiency for different cancer cell line, MCF7, HEC, now 293 header cell. And then again, with this thing, we are able to really try to optimize this kind of thing, and then also create a phase diagram, right? So that here, X is CA, and D star is our cell size normalized with pore size, so that we really can some interesting thing. Again, the key thing here is we identify some critical CA between cancer cell CTC and non-cancer cell white blood cell. So there's a big difference between this kind of critical CA for these two family cells. So with this kind of thing, so that we demonstrate this kind of biophysical CA as a new type of biophysical biomarker to do this kind of separation. If you can really try to find some unique this kind of CA, right? And then so that we can let the CTC to go pass through non-CTC, non-cancer cell can now see, right? So that is something we can really do interesting. By this thing, we discover like critical CA can be used for personalized protocol, so that with this kind of personalized protocol, that means if we get the human blood, we can really try to do personalized this thing for each patient, so that in a P1, P2, P3 is a prostate cancer patient, so that with this kind of personalized protocol, we can really optimize the capture efficiency compared to non-personalized protocol. So that's something quite important. We demonstrate with large number of this kind of clinical study, we can really do much better for this kind of, at least we can detect those cancer stage 2 cancer patient collaboration with those one of the leading cancer center like Dr. Sun Yat-sen University Cancer Center in Guangzhou, and also work with additional medical this kind of center. Actually, we also try to collaborate with Weibo, so see collaborate some of cancer expert in Thailand. So that's something we really demonstrate this kind of technology we very useful, right? So that and of course this can be also used to integrate with what this is based on cell level of this capture those kinds of cancer cell, and we can also continue to look at the molecular detection or seeing to look at this kind of thing, right? And using additional biomarker to look at how this kind of statistic can be useful for personalized cancer diagnosis or personalized cancer treatment. Okay, and again, we also have benchmark with US FDA approved this kind of cell machine made by Johnson & Johnson, so that we demonstrate we can really be better, more reliable, this kind of thing for this kind of clinical research. And then, of course, we actually have been working this kind of from microfluidic device, MEF device, to this kind of electromechanical integration. So in case we are able to do this kind of automation and come with a personalized microfluidic system for practical application with our medical doctors, so that and we have personalized this kind of protocol using computers to do things, and then eventually we demonstrate this kind of CA based thing can really have excellent performance in the capture efficiency, and also depletion of this kind of non-cancer cells, so we can reach some good clinical application, clinical sensitivity, and also we have clinical specificity is 100%. So that's why compete with those kinds of for example, cell search, those kind of real cell in France, and for Void Bioscience, so that we actually have excellent performance. And then of course we continue to make from microfluidic device and with automation, and then going this kind of 4th generation of the system. So that's something I just let you know that we continue to work on this kind of personalized microfluidic device, and then we are in stage to work with additional hospital to clinical study, so that of course this can be very useful, right, for clinical application, so that we are also try to, with help from Weibo, so we'll try to find additional medical doctor in Bangkok and Thailand to see if they can be really useful for more clinical study. Okay, so that I think that we almost finished today's lecture, so that I don't know, do you have any question? Any question for about the lecture? So that again, I believe this kind of microfluidic and also micro nano, this kind of fabricate device biochip can be important for, you can see, for like basic research, for DNA sequencing, for biometric application, even for this kind of a detection of some of this kind of disease, right? So that will demonstrate this kind of microfluidic integrate with this kind of thing can be very useful for this kind of biomedical application. I don't. Do you have any question? Any question for this? My lecture? No. Of course. I would say this kind of semiconductor technology. I would say it's promising because you can see that in the past 60 years, semiconductor has pushed this one to extreme, right? So that I think this kind of semiconductor technology will continue to grow, right? And then so that can be quite interesting thing, right? And Ion Torrent, the founder, has another crazy idea about not only do the DNA sequencing, but also do the what? But also protein sequencing. So that is another crazy idea. I just show you Jonathan Rosenbach, the founder of Ion Torrent, has another company who went to this public in I think New York stock market. So that let me quickly show you one final video about
the future of next generation.
Let me show you the final video about this Jonathan Rosenbach's new idea for DNA sequencing, now protein sequencing technology.
Welcome to the future of next-generation protein sequencing with the first truly accessible proteomics platform. Combining biology, chemistry, and semiconductor technologies, our platform helps see what others can't. Our universal single-molecule detection system generates sequencing information at individual amino acid resolution across millions of independent wells simultaneously. Sample prep begins on Quantum SI's carbon instrument. We isolate proteins and digest them into peptides. Peptides are distributed into individual wells on our chip, which is loaded onto Quantum SI's Platinum sequencing instrument. Each peptide is attached to the bottom of a well. Reagents consisting of recognizers and cutters are added to the chip in a one-pot reaction. Recognizers, each labeled with a fluorescent molecule, bind a terminal amino acid on each peptide. Recognizers bind and release from the terminal amino acid based on affinity and specificity, thereby generating a signal specific to the amino acid present. Signal is generated by fluorescence, as each well provides a reaction chamber in a single molecule detection system. Pulse light stimulates the fluorescent molecule to emit a photon. A sensor measures the number of emitted photons and their time of arrival to compute the fluorescence lifetime instead of color. This is called time domain sequencing. After many binding events of the recognizer, another molecule called a cutter removes the terminal amino acid, exposing the next amino acid in line. A new recognizer binds, and the process is repeated until amino acids in the peptide are sequenced to identify and quantify the original digested protein. This happens across millions of wells on a single chip in parallel. While traditional sequencing utilizes color and intensity to differentiate 4 DNA bases,
the DNA sequencing
this is
will show you
inadequate for the 20 amino
full color.
acids in proteins. Quantum SI combines 3 axes of data to address the expanded alphabet: lifetime and intensity of the fluorescence molecule, along with the binding kinetics of the recognizer. Our chip enables analysis of virtually any biological molecule, including DNA, small molecules, or whole proteins for a truly multi-omics-enabled platform. Join us on a journey to maximize impact, motivated by the idea that our products will change millions of lives.
Okay, so anyway, so this is the final video I just show you, so that I believe this quantum another company from the Jonathan Rosenbach will change the world. And according to our department head in life science in HKUST, has told me that this is once you have protein sequencing is another big problem in biology. So that if this can be true, so that this kind of invention probably can also get Nobel Prize. That's I tell you. This conclude today's lecture. Again, do you have any question? Of course, it's interesting. You may also try to look at this Google and look at this quantum seq, so that this company is just I think just went to public in United States, New York stock market. So that so this is quite amazing. So that okay, so I think we run out of time. So that do any question? If not, probably that's it for today's lecture. Okay, bye bye. You have a question, maybe you try to send the question to Weibo. Okay, then we can try to answer you. Okay, bye bye. Bye bye.